Each organ has a job to perform. Food enters the mouth, is chewed with the teeth, and mixed with saliva, which produces the enzyme amylase, which breaks down the molecules into starch. Starches are an excellent way of storing energy. In the duodenum, a pancreatic enzyme, lipase, breaks down fats into glycerol and fatty acids. Another enzyme, amylase, produced by the pancreas, breaks down starch into maltose, a disaccharide sugar. Trypsin and chymotrypsin are powerful pancreatic enzymes that split proteins into polypeptides and peptides. Enzymes produced by the lining of the small intestine are sugars, maltase, and lactase. They connect disaccharide sugars to monosaccharide sugars. Peptides are also made in the intestine, which splits peptides into smaller peptides and amino acids. Nutrients are absorbed from the lining of the small intestine into the bloodstream, which transports them to all cells of the mind/body, where assimilation occurs. Undigested foods enter the large intestine, where water and salt are absorbed by the intestine lining. The residue, together with waste pigments, dead cells, and bacteria, is pressed into feces and stored for elimination. Enzymes are protein molecules that increase the speed of chemical reactions in the mind-body. They work by combining and altering the molecules of other chemical substances. There are thousands of molecules of different types of enzymes, with varied structures determining the particular activities of all molecules. The digestive enzymes secreted in the digestive tract are responsible for splitting molecules of foods into smaller units for absorption. The nutritional biochemistry and metabolic pathway of the body's natural state, exceptionally where nutrient availability is altered, provide room for disease. The function of the metabolic pathway is to break down complex substances into more simple compounds, for example, protein to amino acids. The reverse process synthesizes simple compounds into a more complex molecular process. The breakdown reactions are called catabolism and anabolism. Catabolism is the breakdown of complex compounds into simpler compounds. This includes the digestion of food into smaller molecules and releasing energy from these molecules within the cells. Anabolism is the building of simple compounds into substances needed for cellular activities and the growth and repair of tissues. Another way of looking at the same reaction is the standard free energy changes that occur during their course of action to drive them into energy yielding or energy using reactions. Overall, as mentioned before, perfect efficiency may not be achieved. However, there should be an energy balance within the cells. This is why a cell never releases two types of reactions in isolation. Energy yielding ones are always linked to energy using responses, and one is used to drive the other. In any one pathway, the two reactions are found to be connected to a common chemical intermediate, which processes a highly reactive group. The intermediate, with its reactive group, is formed at the expense of a small number of compounds ubiquitously present in the cell, existing or being in all places simultaneously. There are three types of such small groups of transfer molecules. The most important of all the group transfer molecules is adenosine triphosphate, ATP. ATP is considered by biologists to be the energy currency of life. It is the energy molecule that stores the energy we need to do almost all activities for life. ATP is present in every cell, and essentially all the physical and chemical reactions that require power for operation are obtained directly from stored ATP. As food in the cells is gradually oxidized, the released energy is used to restore the ATP so that the cells always maintain a supply of this essential molecule. When several sequences of molecules are linked together, they are referred to as macromolecules. The most significant thing about these giant molecules is that they possess a specific individuality that simple substances lack. Two molecules of glucose or ATP are identical to whatever source they are prepared from. Two molecules of copper, sulfate, or water are broken down similarly. However, within the long chains of repeatedly linked units that comprise the macromolecules, there is room for various mistakes or mutations. What's important is the precision and variation in the structure of molecules. One such case is that of hemoglobin, a blood protein that contains over 300 amino acids. If the sequence is changed by having a single amino acid out of line, the hemoglobin won't function properly. This results in the previously mentioned disease known as sickle cell anemia. There are many similar cases of large chains of molecules substituting themselves in any sequence that determine whether we live or die from diseases. Matthew, a survivor of cancer and stroke, requests that you help spread awareness of severe diseases by publishing this video. Please take a moment to subscribe to and like this video.